Let's get ourselves started with topic F2 in this case, which is still continuing with classification, but now we're going to focus on viruses. And then way towards the end, we'll just kind of highlight a little bit of the kind of exceptions or little strange cases, things like viroids, virinos, prions, and things like that. Right? But let's cover what we want to know. As far as classification goes, specifically, we're going to introduce a brand new subject. So yes, we're talking about viruses, but more than anything else, prior to talking about these things, we need to know really what they are. And so by that, we need to introduce what we call the living criteria. As you're going to learn shortly as we go into the next set of slides, we need to know how to define the viruses and how they cause our diseases and so on and so forth. And then we can classify them, which will be part 2 of that. Then we'll get into how do they get in, how do they hijack the system, how do they take over, how they make more of themselves, and more towards the end of the lecture, we'll end up linking how some of these viruses can cause cancer. So that's the overall story of this guy. So starting off with that, first, we kind of have to focus on what we've already learned. And so far, everything we've covered regarding biology and microbiology is that all the things we're talking about are considered to be alive. And what do I mean by this whole alive thing per se? Is that pretty much all the properties that we've discussed, anything from their structure, meaning that everything has some sort of nucleic acid, DNA, that kind of thing, that is enclosed in some sort of small pocket of lipids, what you guys know as your membrane, right? But all of these things we're looking at somehow are able to respond to their environment. They consume nutrients, they process them, they eliminate some wastes, then they grow not only in size, but they also grow in number. And all of these things that we all kind of take for granted are part of those properties of living things. And then comes a virus, which pretty much does none of those. And so one of the things you'll find out is that depending on who you talk to, specifically anybody that is not a virologist or anybody that is not a microbiologist in this case, will tell you, viruses are dead. They're not alive. Well, just to kind of trigger a little bit of some fun thinking, if they're not alive, how the hell are they killing us, right? How are they doing all these crazy things? How are they adapting so fast? How are they taking over? And why are we having so much trouble taking them down? So one of the things this happens for, the reason why we have this issue of are they alive or not, has to do with specifically how we define life. And the problem with defining life is that humans do it. Humans are largely egocentric, right? We like to define things based on us. I like to make the example of saying, well, how do we define a cat or a dog or something like that? And we go, well, that animal has a tail and also has, 4 legs and some weird ears or something like that. When in fact, well, that animal was always like that. We're just compared to the humans that do not have 4 legs, rather we have 2 arms and 2 legs, and say, well, that one has these other things. That's a little bit of an egocentrical point of view. We're defining it based on what we know, as opposed to what is. And so we call that a top-down view, when everything we compare to, when everything we define to is based on us, based on our perspective. What's interesting is the fact that humans haven't been around that long, especially on this planet. So all these other things have existed, and we just decided, no, we're on top of the food chain, so therefore we define everything based on us. So we call this a top-down perspective. And so what happens is when we look at a virus, we go, well, the virus does not breathe, does not respire, does not perform metabolism, does not do these things. It doesn't respond to its environment itself. It doesn't get bigger like most animals do or something like that. And so we just go, well, nope, can't be considered alive. Seems a little bit unfair. So we look at it from what we call a bottom-up perspective. What if we start defining life as probably the smallest thing, the most basic, tiniest thing that can lead to all these other activities, all these other properties, and from there we define life. If we do that, if we change our perspective and we go, well, there's these particles that also have nucleic acid, and they're also enclosed. They're just not enclosed in lipids, they're enclosed in proteins. And then they can kind of take over other cells in their machinery and pretty much just hijack them completely and then just make them do their bidding and make more of them, make them little babies and float around. Seems to be kind of a different way of looking at life. So it really depends on which book you read, who do you talk to, but as far as virologists go, as I'll start giving you more information, viruses are considered to be alive. we define viruses as alive with some exceptions, right? The key concept here is that our viruses are acellular. What do I mean by that? Is that, well, all the cells we've been talking about have, cells. They have this nucleic acid content inside of it, their DNA. Somewhere inside, this nice little bubble of phospholipids. And so that is our typical cell. Well, viruses then would be considered acellular. So it's still life, but they don't have a bubble of lipids. Instead, they're enclosed into this neat little kind of case of proteins. The other thing that we need to add to viruses is that they're pretty much obligate replicatory mechanisms. In other words, meaning that they need to be inside a cell to be able to make more of themselves. Okay, so if we kind of take those two things into what we define into life, then well, viruses are alive too. One of the problems is when we look at life, people like to kind of take like these little snapshots. You kind of take a little picture of something that you're looking at and go, oh yeah, that thing is alive. Because right now when I'm looking at it, it looks alive. When you take a snapshot of a virus, it really is not doing anything. But if you, rather than focusing on just one little moment in time, you look at history of viruses over the millennia, the millions of years too, you can see that these things not only have been around for that entire time, they've been taking over organisms, they've been hijacking them, they've been controlling them, they've been making more of them, they actually evolve, they change, they respond to their environment. And as you probably hear lately, variants appear that are more resistant, they're more infectious, they're more transmissible, and so they can respond quite well. So rather than looking at a virus as just this tiny little thing, under the microscope that is really not doing much, you kind of have to change your perspective. And the reason why we give you this example is, well, for those of you who remember topic F, or F1, if you will, when we talked about our alpha proteobacteria, and we look at things like rickettsias and coxiellas and a couple of the other ones in the proteus, if you remember, these were really, really tiny bacteria. And those tiny bacteria were able to infect other bacteria or other cells. They were intracellular pathogens. Well, these guys are not that far from viruses either. It's quite possible that our viruses eventually acquired these little lipid enclosings and started taking over, right? And we'll get to what lipid encasings are, things like that. So we'll get to some of those, all right? So let's kind of add some new properties there. So yes, viruses are, by definition, for virology, for microbiology, and us, alive, okay? But they can't quite carry out metabolism unless they're inside a host. They require a host also, a cell, to be able to make more of themselves. Now, they don't possess any membrane organelles, so they don't have any ER, they don't have any mitochondria, they don't have any ribosomes, but they can hijack them, meaning they can use them for their will. In other words, they're efficient. They're really neatly packed, and they can use everything from their environment rather than having to carry it themselves. They travel light. Think about it that way. So most of them don't have a lipid membrane around them. There are some exceptions. Okay, so we'll talk about what happens when certain viruses can have a little bit of a lipid enclosure, something called an envelope. We'll talk about those. And what we do know is that, yes, they do not get bigger, although there are some fairly large viruses out there, but they do grow in number. They do make babies, basically. They do reproduce. They do make more of themselves. But what is going to kind of change our perspective on how we teach this subject is that all of what we saw in topic D changes a little bit more. They don't quite follow the central dogma fully. They kind of do some exceptions to them. And so we'll highlight some of those, okay? So before we begin on this, let's start talking about what are the main properties of a virus. So as we continue to kind of define what are the main properties of viruses, let's give you some basic properties. One, obviously, they're submicroscopic. They're smaller than your average bacteria. So you can even call them nanoscopic if you wanted to go with that term, right? So, however, again, part of the description behind viruses, some of them are large enough to rival bacteria. So some of them can get it pretty large out there, mind you. Now, again, we know that they're acellular, so no phospholipid bilayer outside of them. Okay, no membrane, no wall basically, right? But they do contain a little packaging system. Their packaging system is made up of proteins. And those proteins specifically, when you assemble them all together, they get a fancy little name called a capsid or a capsid enclosure. And within that capsid, within that packaging, within that enclosure, if you will, all right, you have your nucleic acids. You have your DNA, or in this case, possibly even RNA. Now, one of the main other things about them is that part of their main thing, their main requirement is that they have to be transmissible. In other words, they have to be able to be passed on by any method typically, but they have to be infectious more than anything else, meaning that it needs to be passed on from one source to another source, but they have to stay on that final source. In other words, they have to remain and not be eliminated by that particular host. And so once we get into topics I and J, and we need to talk about epidemiology and the definitions of what contagious versus infectious is, we'll get into more of those details. And probably the biggest definition that is going to be different for most of you here is really what we use the term virus for. And pretty much in everyday life, everybody just calls it a virus. And that's kind of where everything goes, even currently in our media. But in reality, our organisms, specifically our viruses, have two states. They have an inside and an outside state. In other words, they have an extracellular state and an intracellular state. The extracellular state is referred to as a virion. Okay, so the outside version of this virus, and I hate using the term again in this case, is considered to be a virion. Only when it gets inside the host, inside the cell that it's trying to hijack, that it's usually called a virus. And so in those steps, as we will see much later, what normally happens is that the virus is only considered to be inside the cell itself, whatever it's trying to take over, hijack, or kill. But it's an external structure. It has this little protein coat, its little capsid, okay? And that's the way it transmits. That's the way it infects. That's the part that is needed to pass on from one host to the next one. It requires that little packaging to be able to do so. And as we'll see a little bit later, it's part of its recognition system. Now, some of them, like I said, may have a little bit of a lipid coating outside. We'll deal with that a little bit later. But we do have separate stages behind them. We have a virion stage and we have a virus stage. The virion is while it's on the outside. The virus is while it's on the inside. And if you actually want to refer to your textbook a little bit, your textbook does a quick bit of a table summary in which it compares cells versus bacteria and things like that. So if you get a chance, you can always use that. I have it posted over here in the notes, so you can have an idea of what big differences exist between those two. Now that aside, let's make sure we understand this concept of how we classify them. That's what topic F is about, right? And so for classification, here's when it gets a little bit interesting, right? So here's where we do the whole taxonomy, the whole properties thing. So let's get that started. Part one behind this is that, well, since we weren't really familiar with viruses until relatively recently, okay? This is something that with the last century and a half of information is relatively new information for us, okay? So what we have is that there's a system that developed, an entire committee got together to say, we're gonna work on the classification of viruses. And so in 1966, they got together and they came up as the International Committee on Taxonomy of Viruses, called the ICTV, okay? And so the ICTV is the one that gets to decide where do they fit. What's interesting about this, though, is that the problem is we don't have enough information behind them. So continuing on with the taxonomy, because we don't have enough information about viruses, even though we've been studying them long enough, there's a little bit of a limitation. As opposed to dealing with things like bacteria and some worms and things like that, most people are not willing to volunteer, to get Ebola injected into them just to see how it works, right? So there's a little bit of a challenge in this case. So for that, we have sometimes limited information on how a virus infects, progresses, and so on and so forth. And so as we began sorting them out, as we began classifying them, and including them in systematics and taxonomy, as complex as they got, we only got up to the tier of order. So as you'll notice, we only can classify viruses currently up to order, family, and going down all the way to genus and species, which is the same terms you guys are all familiar with. In other words, you'll notice that there's no class, that there's no phylum, there's no kingdom, and there's no domain. Interestingly enough, as you all remember from regular classification, right, is the fact that we had our three main domains, right, our eukarya, our bacteria, and our archaea. And when I threw that concept of viruses, well, we really didn't know where to fit them. We just didn't have enough information. So in a future lecture, I'm actually going to give you some updated information on what has happened with the classification of not bacterial viruses. And hopefully you'll understand how far we've gotten with that research. And this is as research just as far as 2020, funny enough. Okay. And so when we name them, you'll find out there's a quick little pattern, right? So if they're at the level of order, for example, all of these will end up with virales. If they're at the family level, they'll be called Viridae, Virinae, virus, and then we usually get the name like Ebola virus, or in this case, SARS-CoV-2 or something like that, right? But let's look at how we've classified them. What do we do to get them to that level? So there's three main things that we utilize to classify our viruses. We use more or less their genome structure, so their nucleic acids, that's usually the main one, right? And then we also look at the host, so who do they infect, what do they infect, if you will. And so the reason behind it, there's a lot of range, not only just humans or just animals or just mammals or even just vertebrates, but they can hit plants, they can hit shrooms, they can hit across the board, they can hit bacteria, they can jump between them, as we're learning very recently. And then we also get to look at what their shape is, their overall structure behind their protein, behind their capsid. That's also a way that we can classify them. Now, because they're somewhere between just in terms of mammalian viruses, I want you guys to understand, just in terms of viruses that infect mammals, there's somewhere an estimate between 300,000 to about 1 million mammalian viruses out there. So a lot of them are going to overlap. There's going to be a lot of range in there. And so sometimes those three categories are just not enough. So when those are not quite there, we sometimes use what disease they cause and what type of symptoms and signs they produce. What type of immune system do they elicit? What type of response do they kick in from our own immune system? So their antigenicity. Sometimes what are the proteins that they all produce? So we create what we call a fingerprint of their protein profile. And again, things like host ranges and things like that. So when those three don't quite work, we start using some other subcategories. So anyway, let's start with the very first one. Let's deal with the genome itself, right? And in this case, this is where we get to kind of declare that whole idea of size mattering once again. For those of you remembering from topic C when we described the cell, and then we talked about topic D when we got into the central dogma, we told you that at least humans typically in their cells, and their couple of trillion cells that they possess, each cell possesses roughly about 6 feet worth of DNA in each individual cell, mind you. So again, 2 meters, the same distance we're asking you to maintain social distancing. So we get 6 feet worth of DNA inside one tiny cell. Well, if we take that into perspective, when we look at something like a bacterium, which is significantly smaller, most bacteria will be in the micron size, right? We saw this under the microscope. That's what we were dealing with. What's interesting about that is that most of these bacteria, their genomes are also going to be significantly smaller. And E. coli is about a couple of microns in length, and its DNA, its genome, is about almost 2 millimeters in length. So for those of you who haven't figured that one out, that's about a 10th to a 15th of an inch. Okay, so again, kind of tiny. Now, what you're seeing in the pretty picture over here is the DNA of E. coli. I kind of spread out, but over about a quarter of it. And so you see all that stringy stuff kind of floating around everywhere in there, right? So that's all its DNA. That's a partial chunk of its DNA. Now, just to emphasize, in case you haven't noticed with the pretty picture, over here, where it says viral genome, that's the size of a virus genome. That's its genetic content. And remember, you're only seeing a quarter of that from the pretty picture over there, right? To give you a slightly different perspective behind this, right? Chlamydia trachomatis, as we will learn from the topic F in this case, has roughly between 600 to 800 genes, depending on which species we're talking about. One of the smallest viruses that we know, known as MS2, which happens to be an enterobacteriophage. So this is a virus that infects bacteria, specifically of the colon or enteric bacteria, has 3 genes. 3. That's all it has. So you can see how much in scale, right? There's a difference in terms of how much DNA or how much nucleic acid, it carries. So putting that apart in this case, let's give you more variations. Not only will viruses kind of change our understanding of the central dogma, one of them being in this case, is that not only can they carry DNA, but they can also carry RNA instead. Never both, mind you. As far as we know, no virus at this moment has both of them at the same time. But your virus can be composed of DNA, or it could be composed out of RNA. So there's this classification on RNA viruses versus DNA viruses. But even then, not only can it have DNA or RNA, its DNA or RNA can be in either single-stranded form or double-stranded form. In other words, it can have just 1 strand or 2 strands of either of those two. In other words, it can have single-stranded RNA, single-stranded DNA, and it can also have double-stranded RNA and double-stranded DNA in either one of those options. Even more so, viruses can come in multiple sections. So just like humans and most mammals have chromosomes, right? We divide up our DNA into little chunks. So can they. They can either have one large chunk of their DNA or RNA, or sometimes they can have little divided chunks of their RNA or DNA too. And even more so, similar to humans versus bacteria, they can have it in either linear fashion or circular fashion too. And just to add a little bit further damage to how much information there is in their nucleic acids, they also have a variation in terms of what we call their decodability. In other words, how fast can they make protein? How effective are they at making protein based on their nucleic acid content? So a slightly different version of this, a slightly different picture of this is kind of showing you how many options or variations exist behind these guys, right? Again, DNA or RNA, single-stranded, double-stranded, linear, circular, 1 chunk, many chunks, that kind of thing. All of these are things on how we look at viruses. And I hope you're starting to get a picture of how complex this can actually get, right? And so I was leaving the last one for the best case in this case to describe what is this decodability, something that is a little bit more applicable these days, especially when we start talking about viruses. And so decodability, another way that you'll hear talk about, will be referred to as sense, or known as commonly as positive and negative sense. And so what does that mean? So what I'm going to do is briefly, I'm going to use my little doodler down here so you guys can see more or less a pretty picture of how that works. So the 2 definitions here, so as I move around over here to see if I can do this. So work with me over here, people, is we all know, as we learned from topics C and D, when we looked about the central dogma, is that most of us come together with, our awesome little double-stranded DNA, right? We have our cool little double-stranded DNA, right? And then it could replicate, so undergo replication. I'm just going to put a little R over there. And you can make another copy through semi-conservative replication. We leave that guy alone. But we also learned that through this wonderful little process of transcription, using an RNA polymerase, we can turn that into a single strand of RNA, right? And then even further through the process of translation, I get my colors over here straight. Through using those awesome ribosomes that we had talked about before, right? You'll get this cool little protein, right? Through its primary, secondary, tertiary structures, all that good stuff that we talked about. And those are the guys that perform some sort of function. That's what we learned so far. That was our central dogma, DNA, RNA, protein, right? And so if we follow that path of, again, DNA, RNA, protein, that's usually what we describe as the positive sense. The one that we all kind of silly said makes sense. That's what we know. That's how we understand most of the life out there. Now, what's interesting about it is the converse, the opposite, the negative sense. So let me erase this all over here and just continue to build off of that. The negative sense, what happens is that it usually requires extra steps to kind of produce protein. So let me go back to that DNA again. So let me draw this guy again over here. Here's my little one strand. And I'm going to put my second strand just right below it. And you all remember that there we had a coding strand, and then we also had a complementary strand, right? And it was specifically the coding strand that we usually define as the positive sense one. This is the one that we're going to derive the information from. That's the one we will get eventually our RNA, and that's the one that will eventually get our protein. The negative sense is typically, not always, but typically the other strand. So I'm going to change color for it. I'm going to go with this one. I'm just going to go with a little bit lighter blue over here, if this thing lets me. There we go. All right. This one is what we will consider the negative strand. And it's usually referred to as negative because we can't get protein from it. If we turn that guy into RNA, eventually it's going to give us something weird. It's not going to give us the right codons. It's not going to give us the right information to produce the protein we want to make. So that's usually referred to as a negative sense. And so what this negative sense requires as an additional step is that it needs to be converted back onto the positive sense. In other words, this strand right here needs to be converted into its original positive strand. Trying to get this guy to work really quick. My screens are going a little haywire. You will need to convert it again back to its positive strands, and then eventually you can make your protein. Now, it gets more complex than that. Sure, we can understand the difference between positive and negative, but it doesn't stop there. What's something very unique about viruses is that, sure, they could start as DNA, double-stranded or single-stranded, or again, positive sense or negative sense. But again, remember that our viruses can also start as RNA. And we know that the positive sense of RNA will directly produce protein for us. Okay, but what about the negative sense? Well, again, the negative sense doesn't work. It needs to be converted back. So what happens when we create a negative sense one? I'm going to try and change the color just a little bit. I pretend I have a negative sense one over here, right? Hopefully you guys can tell the colors a little bit more apart. Is that the negative sense RNA cannot produce protein. Instead, it needs to be converted back to positive sense before your ribosome can read it and then turn it into something awesome and functional. So the question then becomes, well, why? Why would you need an extra step to make protein? Why would you even bother with that? And as we'll learn a little bit later, there's a little bit of a switch in there too. Well, one of the things we learn very quickly is that this is a very efficient way of making protein, a much faster way. Why? Because what will end up happening is that, remember, the main requirement is that you have to have positive sense to make protein. And that guy will make more protein and so on and so forth. But the negative sense one, like I said before, needs to make its positive sense first. So let me see if I can select my colors again. Before you can use protein. Okay, fine. How is that any better? Well, you forget that the negative sense is the complementary strand. In other words, it's a template, which then means that we can use that same template, and in this case, make lots and lots of positive strands all at the same time. And now every single one of them can now make us protein. In other words, you made a cool little assembly line of positive senses just from one negative sense version of it. And the same thing applies to DNA, mind you. If you have a negative strand DNA, the negative sense version, you can make multiple copies of the positive sense, and now you can make tons of them as well. So it's a very advantageous system, right? So this is kind of where we will continue to expand on why viruses are kind of difficult to kind of classify, to study altogether, and more than anything else, how to find cures and treatments behind them. They're not as simplistic as the central dogma with a bacterium, or with the worm, or with a fungus, or something like that, in which everything is DNA, RNA, protein. Here, we start seeing our very first examples of issues with, we can't even understand it's DNA or RNA sometimes, depending on what it contains. And that's just one level of their classification, okay? So we'll stop here before we go into our next little topic.